Microbiology for GNM Unit 6, Practical Microbiology Microscope. A microscope is an instrument used to magnify small objects or organisms, making them visible to the naked eye. It is essential in microbiology for identifying bacteria, viruses, fungi, and parasites. Types of microscopes: compound microscope. Has two or more lenses, objective plus eyepiece. Provides higher magnification, up to 1000x or more. Used in labs to view bacteria and cells. Invented by Zacharias Janssen in the 1690s, developed further by Robert Hooke. Electron microscope uses a beam of electrons instead of light. Extremely high magnification, up to 1 million times. Can see viruses, organelles, and fine cellular details. Invented by Ernst Ruska and Max Knoll in 1931. Two main types: TM, transmission electron microscope, internal details, CM, scanning electron microscope, surface details. Parts of a microscope: eyepiece, ocular lens. The lens you look through, usually 10 times magnification. Example, you see the magnified specimen here. Objective lenses, located on the rotating nosepiece, provide various magnifications: 4x, 10x, 40x, 100x. Example, used to zoom in on bacteria or cells. Stage, flat platform where the slide is placed. Example, supports the specimen slide during viewing. Condenser, focuses light onto the specimen. Diaphragm, controls the amount of light. Coarse and fine adjustment knobs, focus the image by moving the stage up, down. Arm and base, used to hold and support the microscope. Uses of a microscope to observe microorganisms, bacteria, fungi, protozoa in clinical samples. Example, identify plasmodium in malaria. To study cell structure, RBCs, WBCs, epithelial cells. Example, observe red blood cells in anemia. In labs and diagnostics, used in microbiology, pathology, and hematology. Example, detect pus cells in urine microscopy, handling and care microscope. Handling tips: carry it properly, use both hands, one on arm, one under base. Example, prevents dropping and damage. Start with low power, begin focusing on 4x or 10x objective. Example, helps find the specimen easily. Clean lenses gently, use lens paper or soft cloth only. Example, avoid scratches and dust build-up. Always cover with a dust cover when not in use. Keep in a dry, clean place. Don't touch lenses with fingers. Staining, slide preparation and microscopy. Slide and smear preparation. To fix microorganisms onto the slide for staining and microscopic observation. Clean the slide, use alcohol or flame to remove grease. Example, ensures clear viewing. Prepare the smear. Place a small drop of specimen, e.g. pus, sputum, blood, on the slide and spread it thin. Example, allows even staining. Air dry and heat fix. Let the smear dry and pass it quickly through a flame 2 to 3 times. Purpose, kills microbes and sticks them to the slide. Staining procedures, basic. To make microorganisms visible under the microscope by colouring them. Gram staining, differential stain. Steps, crystal violet, primary stain. 1 minute iodine, mordant. 1 minute alcohol, decolorizer. Few seconds, safranin, counterstain. 1 minute, results, gram positive, purple, e.g. Staphylococcus aureus, gram negative, pink, e.g. E. coli. Use, identifies bacteria type and guides antibiotic therapy. Acid-fast staining, Zeal-Nielsen. Steps: carbylfusin, heat for 5 minutes, acid alcohol, decolorizer, methylene blue, counterstain results, acid-fast bacilli, pink-red, e.g. Mycobacterium tuberculosis, non-acid fast, blue use, TB diagnosis, lactophenol, cotton blue, LPCB, stain, use for fungi. Result: blue-stained hyphae and spores. Example: Aspergillus species, examination under microscope. Place the stain slide on the microscope stage. Adjust light and focus. Start with low power 10x objective to locate the smear. Move to high power 40x for closer look. For bacteria, use oil immersion 100x lens. Add a drop of immersion oil on the slide before focusing. Observations to record, feature, what to look for, shape of bacteria, Kochii, round, bacilli, rod, spirilla, spiral. Arrangement, chains, e.g. Streptococcus, clusters, e.g. staph. Stain reaction, gram plus, purple, gram minus, pink. Background, clear or stained depending on method. Identification of common microbes. Under the microscope, microbial morphology, microscopy, helps in identifying microbes based on their shape, size, arrangement, and staining characteristics. Common methods include gram staining, acid-fast staining, and wet mount examination. Bacteria, morphology under the microscope, shape, name, examples, arrangement, staining technique, spherical, Kochii, Staphylococcus aureus, singles, pairs, Diplococci, chains, Streptococci, clusters, Staphylococci, gram stain, rod-shaped, bacilli, Escherichia coli, Bacillus anthracis, single, pairs or chains, gram stain, comma-shaped, Vibrio cholerae, single curve, gram stain, spiral-shaped, spirilla, spirochetes, Treponema pallidum, spiral, corkscrew, dark field microscopy, club-shaped, coroniform, Corynebacterium diphtheriae, Chinese letter arrangement, Albert stain. Fungi, morphology under the microscope. Type examples: morphology, yeasts, Candida albicans, round or oval cells, budding. Molds, Aspergillus, Penicillium, hyphae, branched thread-like structures, spores. Dimorphic fungi, Histoplasma capsulatum, exist as yeast at body temperature and mold at room temperature. Viruses, morphology under electron microscope. Examples: shape, structure, visualization. Method: influenza virus, HIV, herpes virus, spherical, icosahedral, helical. Envelope, capsid, nucleic acid core, electron microscope, only too small for light microscope. Note: viruses cannot be seen under a regular light microscope due to their extremely small size. Gram staining differentiates bacteria into gram-positive, purple, and gram-negative, pink-red. KOH preparation helps dissolve debris to visualize fungal elements. Giemsa stain is used for blood parasites. Wet mounts are used for observing motility and fresh morphology. Electron microscopy is used to study viruses in detail.